Hi, it's Miss Vitale. This podcast is on the history of life and is meant for AP Biology. The fossil record has provided us with evidence for evolution for centuries. Fossils 3.5 billion years old hold the earliest evidence of life. Earth formed about 4.6 billion years ago, but was a very volatile, inhospitable place. The atmosphere was thick with poisonous gases, and the oceans were like hot soup. In the 1920s, a Russian chemist, Oparin, along with British scientist Haldane, hypothesized the conditions on early Earth could have formed organic molecules. In 1953, Miller and UREY created an apparatus to test this hypothesis. They used blown glass and they created a chamber with gases that were thought to be in the early atmosphere. They inserted electrodes to create sparks simulating lightning. They heated a bulb of water at the bottom so water vapor moved through the apparatus. After a week, amino acids, hydrocarbons, and other organic molecules had formed. This type of experiment demonstrates that organic molecules can form from inorganic substances. However, this is not even close to the formation of a living cell. Think of the hot, bubbly soup that was the ocean on early Earth. Suppose inorganic compounds such as amino acids were present, and that hot mixture washed up onto rocks. Evaporation would actually cause dehydration synthesis, and proteins could form from amino acids being washed up on rocks and the water evaporating. This phenomena has been shown in experiments. In order for something to be considered living, it must use energy for metabolism and reproduce. Protobionts are abiotically formed molecules surrounded by a membrane. They display some basic properties of life. Again, lab experiments have created protobionts from organic molecules. When oil, which is a lipid, is dropped in water, it forms a small bubble. This can act as a simple plasma or cell membrane. As you know, all living things contain DNA. However, the first genetic material was probably RNA. It has been discovered that certain RNAs can act as enzymes. We call these RNAs ribozymes. In the presence of free nucleotides, ribozymes can copy RNA. The RNA molecules that get copied the most will get passed down more. This is natural selection at work. If this RNA found its way inside a lipid bubble, it could potentially create daughter cells. Eventually, this RNA could have produced DNA, which is a more stable nucleic acid. As we know, the fossil record is some of the oldest evidence of evolution we have. Since fossils form in sedimentary rock layers called strata, it is an actual timeline of this history of life on Earth. Radiometric dating uses radioactive isotopes that decay at a constant rate regardless of heat, pressure, or any other natural processes. The rate of decay is expressed as half-life. That is the time it takes for 50% of the parent isotope to decay. Living organisms accumulate radioactive isotopes, but when they die, those isotopes stop accumulating and remain fossil. The rock surrounding the fossil can also be used to determine the approximate age of the fossil. Characteristics that are unique to certain animal phyla, such as bones and teeth, can easily be used to identify a type of animal. The history of life on Earth is divided into 3 eons. Most of the animal life has existed in the most recent eon, the Paleozoic eon. Eons are divided into eras whose boundaries are defined by mass extinctions. The oldest fossils are stromatolites. They are created from prokaryotes binding layers of sediment together. These fossils are 3.5 billion years old, but weren't the first organisms. They probably began much earlier. We do know that for close to 1.5 billion years, prokaryotes were the only organisms that existed. As you know, oxygen wasn't present in Earth's early atmosphere until photosystem II evolved and the splitting of water produced oxygen gas. However, initially the oxygen dissolved in the oceans until over time the concentrations got high enough to diffuse into the air. This probably happened close to 3 billion years ago. A little over 2 billion years ago, oxygen levels increased dramatically and changed the course of life on Earth. The rise in the oxygen levels probably killed a lot of the existing prokaryotes. Cellular respiration evolved, which was followed by the evolution of eukaryotes. Photosynthetic eukaryotes further increased the oxygen levels. The oldest eukaryote fossils are 2.1 billion years old. Endosymbiosis is the theory that mitochondria and chloroplasts evolved from prokaryotes that were either engulfed by another cell or that parasitized another cell. Serial endosymbiosis states that mitochondria evolved first and chloroplasts evolved later. Endosymbiosis is supported by evidence that includes similarities between mitochondria and chloroplasts and certain prokaryotes. Some of those similarities include similar DNA and ribosomes. As single-celled eukaryotes diversified, multicellular forms arose about 1.5 billion years ago. However, they were small and relatively simple. Larger and more complex eukaryotes didn't emerge until about 565 million years ago. This corresponds to the end of a major ice age. Early in the Cambrian period, one large burst of evolution occurred, the Cambrian explosion. From 535 to 525 million years ago, animal phyla that included hard shells and predator-prey relationships appeared. Remember, life began in the seas, and up to this point, it did not exist on land. That began about 500 million years ago. Plants and fungi with adaptations to survive on land colonized at the same time. Tetrapods, which are animals with 4 limbs, evolved from lobe-finned fish about 365 million years ago. And Homo sapiens have only been around for about 200,000 years.